So this project was initiated by L'Oreal Early. She's a talented postdoc who was working at University of North Carolina at the time and sought to partner with us on a PANC2 gene therapy. She initiated the project and took it forward for the first year and then elected to take a different career path. And so we have picked up the project in our laboratory and carried it forward. This work is supported by the 3 family organizations and donations made through those organizations. And the argument for doing a gene therapy project is to hedge our bets, if you will. It's to invest in a number of complementary candidate therapeutics and develop them in parallel so that we are being as fast and efficient as possible and trying to promote the best treatment. So this is all just proof of principle work. It's very early in the work, but we did want to make the community aware of the work. So L'Oreal built 3 constructs, all containing the full-length human PANC2 cDNA. This is the main one that we've been working with so far in the work we've been doing, the very early work we've been doing. But we do have other constructs and plans to be able to use these all in AAV9. So in this work by L'Oreal, she's showing in Western blots that the lysates from cells transfected with the plasmid containing these constructs is expressing PANC2, both in the mature and in the precursor forms, and also can be detected with an anti-flag antibody as well as antibodies to the human protein.
Susan,
Dr.
I don't
Hayflick?
think people are seeing your screen.
Oh.
Yes, please share your screen, Dr. Hayflick.
Okay.
Thank you.
Now you can see my screen. Sorry. I'm just going to go back, show you the constructs, and then show you the Western. So again, the precursor and mature forms, and then the anti-flag antibody. And then she transfected HeLa cells with these plasmids, the virus containing the constructs, and shows that they co-localize in presumably mitochondria and emerge with the flag, show that we're detecting the protein's getting into the cells. So one of the distinct aspects of this project as compared with other gene therapy projects is that we're not intending to target the entire brain. And so the science of stereotactic injection and the other gene therapy trial plans using stereotactic injection have advanced to a stage where we think this is actually quite feasible. And so this is the framework in which we've developed this project in the mouse. This is just showing you the atlas from the Allen Brain Institute and the target sites that we were aiming for. So all of these injections were done by Shelley Wynn at OHSU, initially in conjunction with Dr. Early. These just using a GFP expressing construct and showing that they were able over a course of 5 to 7 animals to consistently hit the GP or the GP region. And so we developed just an experimental plan with the injection, a period of delay to allow the animals to both recover, to be observed, and also to allow for maximal expression of the proteins, of the PANC2 protein, and then with analyses to follow. So again, it's very early data. So these are human cell line controls from a person with PKAN and a healthy control. These are the mouse data from the GP, our target, versus the cerebellum, which is outside of our target. And then this is a sham injected animal. And these are two animals that were injected with the AAV containing cargo. And one of the consistent things we're seeing, we've got a broader data set than this, but one of the things we're seeing consistently is really pretty significant variability. Maybe this is commonplace in the gene therapy world, but we're seeing quite a lot of variability in the amount of expression we're getting at the same time point. So all of the animals were injected on the same day, sacrificed on the same day, but we're seeing quite a lot of variability in expression levels. But we are seeing the protein now expressed. So this is a knockout animal. The red is nuclear stain, and then the brown stain is with an anti-PANC2 antibody. And we are seeing the antibody detect protein now. This is just a magnification of this cut here, telling us that the injection is targeting the right region. So this is globus pallidus, that's injecting into the right region. The virus is able to transfect cells, and the protein is able to be synthesized. So that's as far as we are in the work. There was a long hiatus after L'Oreal dropped out of the project, but the family organizations led by AIS and AF were generous in supporting us to carry the work forward. What we intend to do is to demonstrate that we can, with the injections, consistently get a high level of protein expressed in these regions, in the target region, and then using the same paradigm of biomarkers and disease phenotype that Dr. Jiang spoke about earlier this morning, looking at some of those markers. So looking at CoA synthase gene expression, complex 1 activity, and PDH, and some of those others. So with that, I will acknowledge the people I worked with here, actually the people who do all the work, and close. And then I'm happy to try to answer questions. So ODC Bone asks, do you think you can rescue the testis phenotype with this approach? It's an interesting question. So what we know about the testis phenotype, we've actually done quite a lot of work studying the testis phenotype and have not yet published it. We do have a paper that is just about on the edge of being ready for publication. There is very early apoptosis of the spermatogenic precursor cells. So it would have to be done quite early. It may even need to be done prenatally, but it's a good thought. And since we're injecting and sacrificing the animals, that might be a good parallel part of the experiment. So I like that idea. Fatima asks in the chat, have you observed any symptoms improved after the vector construct injection? These animals, the knockout animals, do not manifest an overt clinical phenotype. However, as I mentioned earlier, they have a remarkably aligned biochemical phenotype that recapitulates the disease in humans. So we see iron dyshomeostasis, we see dopamine dyshomeostasis, and we see GP-specific disease. I think those aspects of the phenotype, which we won't see clinically, but we will see biochemically, those aspects of the phenotype are very compelling. A few more questions. Does endogenous PANC2 expression change with age? And does this form a problem? Interesting question. I don't know the answer to that question. I don't think we've seen variability, but we have not formally examined that. I'll open the floor in case anybody else, any of the other scientists have. You got to be quick. And then Manju asks, do you think neuronal loss and timing of injections is an issue? In other words, is there a therapeutic window? I do. So I think PKAN begins prenatally. I think that there's a cellular defect prenatally from early in the life of the fetus. And so I would say earlier is better. Beyond that, I'm not sure.